Introductions are normally a personal things, the better you know the people. Right now, I know Jake now since 2004, I guess. That was when you took the geobiology course on Catalina Island, the Agaron course. And in the meantime, Jake has become a professor at the University of Minnesota, the Earth Sciences Department. He started, however, at the University of Arizona as a geologist, made his BS there, and then became a PhD in 2008 at the USC and a PhD in geobiology. When he started there, that did not exist yet, but based on the Agaron course, that became then a field. And ever since, he has combined geology and biology beautifully. Also, his PhD topic was still very geological. Since then, he has become more a biologist. Then in 2007, he took the microbial diversity course in Woods Hole. That's a clear sign that he moved into biology. And then in 2009, after he finished his PhD, he completely emerged in molecular microbiology by doing a postdoc with Victoria Orphan at Caltech. In 2010, he became professor at the University of Minnesota, where he has been ever since. Now, when I looked back at his work as a PhD student, he already connected very closely to what he's doing today. Today, he's interested in the Thiomargarita and the larger sulfur bacteria, but already then, he was interested in large bacteria and in connection with mostly with their phosphate accumulation. He has received many fellowship awards and also has been funded rather well for what he's doing. At the moment, he still gets funded heavily by the Simons Foundation. That's the same foundation that also funds our course. He's a geologist by training, I said, but he works also together with the dental school at his university. And he might say more about that, how geology and dentistry move together. But today, Jake will talk about possible symbiosis involving the world's largest bacteria. One last word. Without Jake, we would probably not have been able to do the RGNO course because he has, since its beginning, he has been a stronghold coming every day, every year, again, participating in the course. And mostly thanks to the fact that Thiomargarita cannot be cultured anymore. I hate to think what will happen, Jake, if you ever succeed in culturing them in your lab. Okay, with this, it's up to you now, Jake.
Great, thanks, Kurt, and thanks so much for having me, and thanks for having me as part of the RGNO course for so many years. It's really been great, and I hope we have many more years ahead of us. So today I want to talk about, as Kurt mentioned, I kind of got interested initially in Thiomargarita and their role in basically the precipitation of phosphorus minerals, but I'm going to talk about something quite different today. So a couple of my keywords here are sulfur oxidation, denitrification, chemolithotrophy, parasitism, eukaryogenesis. These are terms that you might run into throughout my talk, and hopefully I'll be able to break down what some of those are and what they mean. Much of the work that I'm going to be showing you today was produced by Dr. Beverly Flood, who's a joint lead investigator on this project, and Mr. Dalton Lepridge, who's a PhD student who will be giving kind of a second part to this, showing some of his specific work that he's been doing for his PhD. So many of the elements that are involved in Earth's surface processes are cycled through both non-biological and biological reservoirs of the Earth system. Here's this little cartoon of the carbon cycle showing kind of all the movement of carbon throughout different spheres or reservoirs of the Earth system. And this term geobiology or geomicrobiology refers to basically the idea that we now know that microbes really are involved in many of these different transformations of elements that are involved in these various different biogeochemical cycles. And many times, but not always, these transformations involve the precipitation or dissolution of minerals. And geobiology is a kind of an umbrella term that looks at interactions of the geosphere and biosphere broadly. And geomicrobiology is kind of a subset of that that looks more specifically at microbes and oftentimes the relationship between microbes and minerals. The organism that we're going to be talking about today is a really exciting microbe or a group of microbes that are known as Thiomargarita. Thiomargarita namibiensis was first discovered off the shelf of Namibia and by Heide Schultz-Voigt, who we have here on the Zoom call today. And Heide did lots of the kind of foundational work with Thiomargarita, including showing that Thiomargarita can be involved in the precipitation of calcium phosphate mineral deposits, such as, i.e. the phosphorites that you heard about in earlier talks. And so the name Thiomargarita namibiensis means sulfur pearl of Namibia. And you can get a sense for that here by seeing this kind of these bright pearl-like strings here in the sediment. These are all chains of Thiomargarita cells. And these cells are really, really massive. They're the largest known bacteria. They can get up to basically a millimeter in diameter. At least the ones I've measured have been a full millimeter, the largest ones for Thiomargarita. And these organisms, they use hydrogen sulfide as an electron donor, or oxygen and or nitrate as an electron acceptor. And basically what they're doing with this is just how in our respiratory chain, we take electrons from food and we pass them to oxygen, and we pump protons and then convert that chemiosmotic potential or charge separation potential into ATP to power all different types of cell processes. These organisms do much the same thing, but they use reduced sulfur compounds as the electron donor. And here this could be oxygen or it could be nitrate. And really it's actually more complicated. There's these organisms have many biochemical pathways to metabolize a whole variety of different sulfur compounds. But the basic idea is that they're using reduced sulfur compounds as a electron donor. But there's also evidence that they can use other things as electron donors as well, things like dihydrogen, for example. These organisms are extremely large, but much of the solid volume is actually occupied by aqueous vacuole that's been proposed to contain nitrate. And also these organisms contain lots and lots of different storage compounds, including polyphosphate, which is basically this kind of, you can think of it sort of like the phosphate chains on ATP, but many hundreds or thousands of residues long that actually makes this kind of granule that's in the cell here. And these organisms have a variety of diverse kind of morphologies and also a complex kind of biogeography. So here's some examples of different places where Thiomargarita and Thiomargarita-like relatives are found on many different shelf environments, as well as methane seep type environments. And you can see there's a variety of morphologies here. And these organisms are also basically within the Vegetaceae. They're a kind of a clade to themselves, but they're closely related to organisms like Mari Vegetoa and Thioploca. So here, Vegetoa and Thioploca, they're kind of similar to Thiomargarita with respect to their major metabolism. But one thing that's quite different is that they are able to, they have gliding motility. So they basically can migrate through the sediments in order to find the interface is where they have access to sulfide and oxygen, or sulfide and nitrate. And one of the things that Haida initially pointed out, and it's basically something that everyone that kind of views Thiomargarita through the microscope can see, is that there appear to be lots of organisms, microbes, that are actually attached to the exteriors of Thiomargarita cells. And you can see here all these kind of obvious big filaments here, but really there's oftentimes a thick kind of mucus sheath around these cells, and oftentimes that sheath is teeming with a variety of microbes, which could even in some cases include eukaryotes, or does include eukaryotes. And so we think of this concept of a microbiome, like, for example, the human microbiome, all the microbes that are associated with humans. We have these diverse communities that are connected with us as multicellular organisms. But Thiomargarita is so large that we think that it has its own microbiome. And so we kind of set out to look at or do some molecular characterization of this kind of microbiome to get an idea of what microbes are associated with Thiomargarita. And so we collected some basically filaments, different Beggiatoa and Thioploca filaments, and as well as Thiomargarita chains. And we basically kind of picked them out of sediment and pooled them. So about 100 or so chains, we pooled them as basically a sample. And then we did what's called amplicon sequencing, where we basically extract the DNA and then amplify a portion of a marker gene, in this case, the B4 hypervariable region of the 16S ribosomal RNA gene. And then we sequence those amplicons, and then we basically classify them and analyze what are known as amplicon sequence variants. And then we basically kind of compared the results of looking at these amplicon results with the communities that were present in the sediment core itself, the sediment with the Beggiatoaceae removed, and then also just looking at the communities of these pooled filaments. So the blue dots here are the filamentous Beggiatoaceae, like the Beggiatoa and Thioploca, and then the green dots are the non-filamentous Beggiatoaceae. And so one kind of thing to point out here is that we pooled these groups based on morphology, right? So there's some limitations there. We don't have like the specific, for example, like we can't pool all the Thiomargarita namibiensis per se. We're basically pooling on morphology. But you can see here that as we look at major taxa of bacteria, that certain ASVs, which are these amplicon sequence variants or sequences from our amplicon sequencing, are enriched in particular groups relative to the sediment. And in particular, there are about 32 ASVs that were really statistically had strong support, that were enriched at a couple of stations where we had enough samples to do good kind of statistical comparisons. But there are also a whole number of other bacteria that have kind of not necessarily, you can't say statistical association, but also what appears to be a strong kind of association with non-filamentous Beggiatoaceae, the Thiomargarita. Okay, so the groups, kind of some of the major take-home points. There's lots of different taxa there, but we had some of the planktomycetes, but not the ones that are known to be involved in anammox. We have representatives of the Bacteroidota. These are organoheterotrophs, lots of nanoarchaea, and these are kind of interesting, very small kind of parasitic epibionts of Ignococcus, or they're known to be, which is another archaeon. And so they're known basically to have kind of interactions with other cells, other symbiotic interactions. And all these organisms here, they're a lot of them are basically known kind of heterotrophs or fermenters that could be involved in possible kind of syntrophic interactions with Thiomargarita, either providing substrates or vitamins or whatever, or receiving them in some cases. Also, we have sulfate reducing bacteria from the Sulfobacteriota and sulfur oxidizing Campylobacter. So we have a whole diversity of organisms that are associated with our non-filamentous Beggiatoaceae. So you can see here, there are lots and lots of organisms that seem to be kind of closely associated with our Thiomargarita-like bacteria, and that are basically very enriched over kind of the background sediment. So that's kind of a picture of what we think some of the organisms are that are associated perhaps with the exteriors of Thiomargarita. But we also have been working for some time on kind of an interesting, what we think quite interesting observation of cells within Thiomargarita as well. For a number of years now, looking under the microscope at Thiomargarita, we've noticed that there are what appear to be little cells moving around, little bodies moving around within many, many Thiomargarita cells. And you can see here, this is a picture. So these are sulfur granules within Thiomargarita, but then that stuff that's kind of moving around, these are, we think, bacteria. At first, we had a number of hypotheses that basically the host cell was just dead and decaying, and maybe these were just bacteria that were consuming the dead Thiomargarita cells, or that they were maybe infected by giant viruses. Giant viruses are known now that get up to the size of bacteria. Maybe they were actually some kind of reproductive form, so like baeocytes or spores or something of Thiomargarita. Or the other possibility is that they have one or more endobionts. And this would be interesting for a number of reasons. Bacteria are typically not known to have endobionts. There are a couple of published exceptions to this. There are these insects called mealybugs that basically have beta proteobacteria living inside of their cells. And inside of those beta proteobacteria are gamma proteobacteria. So it's kind of like these Russian dolls, kind of nested symbiosis. But the host organism is still a eukaryote. So there's a eukaryotic cell, and then a bacterial cell, and a bacterial cell inside of that bacterial cell. There's also some interesting work by Larkin and Hank that showed actually some TEM work. And this is from the 90s, and they had some TEM of sulfur oxidizing bacteria, basically Beggiatoa, that are, as I said, related to Thiomargarita, that they suggested had basically endobiont bacteria in them based on these kind of TEM images. And so we actually did some TEM, which is very challenging, and we're still working on doing additional TEM, but doing TEM with Thiomargarita can be quite challenging for a number of reasons. But here, this is basically a TEM image of, here's the exterior of Thiomargarita, and here basically is kind of the wall of Thiomargarita, and the interior cytoplasm, and all this stuff here, these are the endobionts here. And you can see there's a number of cells here, and they have, for example, inclusion bodies in them and so on. And there's, looks like maybe a couple of morphotypes or one morphotype with different kind of cross sections here. So that kind of TEM image shows that they're in fact inside the cell. Here we did a number of approaches to look at the question of whether or not they're actually alive. So things like a live dead stain here. So this is the edge of Thiomargarita cell, and this is confocal microscopy, and all those little bright green dots are the endobionts. inside of it moving around. And this also shows that they're inside because it's a confocal image. Things like tetrazolium redox dyes that basically are reduced by the electron transport chain, basically in actively metabolizing organisms, show that these cells that are hosting these endobionts are actively metabolizing. There's also, I'll mention this again a little bit later to explain what they are, but these organisms can take up fluorescent amino acids. So both the kind of host cells on that sheath, so you can see all these are cells in the sheath with the yellow arrows pointing to, and then the white arrows are pointing to cells inside of the host Thiomargarita that are taking up these fluorescently labeled amino acids. Also, we see apparent kind of inheritance of the endobionts across division planes, right? So we see like the dividing cell here, and we see this mirroring of the cloud of endobionts across this division plane. So who are these endobionts? You may be familiar with the idea that there are a number of animals that have sulfur oxidizing endosymbionts that are, many of them are gamma proteobacteria. And examples include the seboglinid tube worms. And these seboglinid tube worms have bacterial symbionts. And if we look here basically, this is a phylogenetic tree showing some of these, the phylogenetic relationships of these sulfur oxidizers that occupy these seboglinid tube worms, including the frenulates and the vestimentiferans down here. And the endobionts that are associated with Thiomargarita are shown here. So they're actually very closely related to known endobionts of these tube worms. And we did kind of a step further after we had kind of the identity of these organisms to show that they're actually the endobionts are within Thiomargarita. So we did fluorescence in situ hybridization, where we basically have these probes for particular groups of interest, these oligonucleotide probes that have a fluorescent tag on them. And then basically that oligo hybridizes to a particular region in the ribosome if it matches that particular group, and then it causes those cells to fluoresce. And here's basically a movie that shows this whole big surface that you're seeing is the edge, the margin of a host kind of Thiomargarita cell. And then, so here's kind of the sheath over here, and this is the host Thiomargarita cell. And then the red structures within it are the endobionts with the fish probes for that particular group of organisms. And so this also shows the kind of position of the endobionts within the host Thiomargarita cell. We also did metagenomics, where basically we extract out the DNA and we sequence all the DNA from the whole kind of community. And then we take those reads and we assemble them and bin them. And then we have basically metagenomes that represent the endobionts that show basically genes for denitrification, sulfur oxidation, carbon fixation via the Calvin cycle. And also, interestingly, genes for the synthesis of type 1, type 2, and type 4 secretion systems, which are basically systems that allow for the kind of the transfer of proteins and targeting of other bacteria or eukaryotes for things like infection. And these organisms are interesting in that they have many kind of similarities in their metabolism to Thiomargarita. So it's one possibility. We don't know if this is the case yet, but it's possible that they're kind of stealing this nitrate, the nitrate that Thiomargarita has if they're denitrifiers. But there are still lots of open questions, like how are the endobionts getting into Thiomargarita? Are the endobionts interacting with Diamargarita? If so, how are they doing that? Are the endobionts influencing the environment? And what is the kind of evolutionary significance of the relationship between Diamargarita and endobionts? So there's lots of open questions, but I just want to spend a couple more minutes kind of setting out one of the other interesting aspects of this kind of system here that will kind of lead into Dalton's talk. And that's that in most kind of models of eukaryogenesis or eukaryoevolution, organelles are thought to be really kind of an important aspect of this kind of evolutionary process, right? And the kind of the canonical kind of picture is that you had some host cell that was engulfed by, or you have some kind of future endobiont that was engulfed by a host cell, or that could have infected them, right? And eventually that that endobiont became the mitochondrion, or in the case of phototrophs, chloroplasts. But it's still kind of there's lots of questions about how that original host cell kind of engulfed that or internalized what would become later on those organelles. And so a lot of the models for how we look at bacteria living inside of other cells come from eukaryotes, because endosymbioses are really common in eukaryotes that can engulf and digest food and do things like phagocytosis, taking up food particles from the environment and so on. Also, eukaryotes have like complex cytoskeletons and so on that can be the targets of infection, can be manipulated by invading bacteria. But there really aren't so many models of endobionts in bacteria. And we don't know if there are, if it's possible. I mean, the kind of canonical view would be that bacteria don't have, they have this kind of rigid cell wall that doesn't allow them to kind of engulf other bacteria or otherwise kind of take them up. And that's kind of our view of the ultrastructure of bacteria, is that it doesn't kind of permit that kind of thing, nor do they, we think, have such a, kind of a complicated enough cytoskeleton to allow such a thing. But we've been doing some interesting work with these D-amino acids, which I mentioned earlier. And these amino acids, or amino acids generally, occur in D and L chiral configurations. And D-amino acids are basically discriminated against by the ribosome's translation machinery. So you don't find D-amino acids in proteins that are made by ribosomes, although they can be made by non-ribosomal peptide synthetases. And so, but one place where you do find these D-amino acids are in the peptidoglycan in the cell wall. And so basically you have the glycan strands in the peptidoglycan are cross-linked by peptides that can contain these D-amino acids. And so if you can put a fluorescent tag on these D-amino acids, which other researchers have done, then you can basically use those amino acids as kind of a tag for tracking the synthesis of peptidoglycan. And that's been done in a variety of different organisms. And you can see some pictures of it here. And so we started doing this with the idea of kind of looking at cell division in Diamargarita, but we found also some interesting features, which we think are basically vesicles that are bound by peptidoglycan. And so you can see some of these vesicle-like features here that are actually inside of Diamargarita. So here's another, this is a kind of a confocal view of some 3 different kind of slices through these cells. And you can see here, these are the structures I'm referring to. And they actually look like kind of an invagination of the cell wall in many cases, that then kind of blebs off these vesicles inside the cell. And you can actually see we think evidence for that in TEM views as well, where you see this kind of enfolding of the cell wall. And some of these invaginations have things like sulfur granules within them, and others are kind of empty. But All this is something that Dalton is going to talk more about. But also just one last thing to point out, in this TEM view I showed you before, you can see that there are actually these kind of thin electron dense vesicle-like features around pretty much most, if not all, of these cells if you look closely enough. And so that's kind of an interesting feature. So there's still lots of open questions. How are these vesicles forming? Do they have a physiological role? And is internalization of however it's happening associated with these vesicles? And so I want to thank the Ministry of Fisheries and Marine Resources, especially Anya, Dion, and Richard, and crew of the Mirabilis. And next, we're going to have a presentation by Dalton Lepridge. And Dalton is going to follow up on a lot of these themes. And so you may have questions for me, and we could address maybe a question if it is, if you want, about my particular portion of the talk, but I might also suggest that we go on to Dalton's portion, and because many of the themes are so closely related, maybe some of his material might help address some of the questions that are out there.